So in our last video, we got introduced to Mendelian genetics, and on this slide I just have a quick review of some of the key terminology that I covered in that previous video. So we talked about how alleles are the different forms of a gene, and those are the discrete units described by Mendel. They're also what's encoded in DNA, and we usually represent them using a capital letter for a dominant allele and a lowercase letter for a recessive allele. We also talked about genotypes, which are the specific allele combinations that can be found in an individual. So each individual that is diploid will have two alleles. So for the case of a gene where there's a dominant and recessive allele, the genotypes that are possible are big S, little s, or little s. So there are essentially three different potential combinations: homozygous dominant, homozygous recessive, or heterozygous. We also talked about how we can't actually directly see those combinations. Instead, what we see is referred to as the individual's phenotype. So, for example, we talked about Mendel's pea plants, and in the pea plant experiments he conducted, he observed the outcome or the phenotypes of smooth or wrinkled seeds. We also saw how Mendel structured his experiments across multiple generations. So he will refer to the parentals as the initial unit that's crossed, and then past that the next generation is referred to as the F1, and then beyond that the F2. We talked a bit about conducting a test cross and a back cross, so methods for breeding individuals to figure out one individual's genotype. So in this video, we're going to take these ideas a little bit further. So the first thing we're going to consider is this example. So what if Mendel had actually started with snapdragons instead of peas? So here are pictures of two different phenotypes of snapdragons. And so we've got a dark pink snapdragon, and then a white snapdragon. So for this experiment, we're going to cross two pure breeding snapdragon varieties, one of these red snapdragons with white. So using the same notation that we'd used previously, we'd represent that as something like big R crossed with little r. So if we do this, what we discover is we go from parents that are red and white to an F1 generation that is 100% pink. Okay, so what happened here? Is this blending now all of a sudden rather than Mendelian inheritance? And so here again, this is one of these cases where it will be helpful to see what happens in the next generation, right? So if we now cross pink snapdragons with pink snapdragons, right? So under Mendelian inheritance, we'd expect these F1 individuals to be big R. So now if we cross pink with pink, if blending is the case, we would just expect to get pink through infinity, right? If Mendel's mode of inheritance is correct, then what we're looking at is crossing big R with big R. And so this is the classic case where each parent can produce two different types of gametes, right? So if we wanted to draw out a Punnett square, we draw out on a 2x2 Punnett square, right? So big R or little r, big R or little r. And hopefully you can start to recognize the different types of Punnett squares that are very common, right? So under this, for thinking about the phenotypes or genotypes of the offspring, then what we'd expect to get would be 1 big R to 2 big R to 1 little r. And as I've just indicated here, if you can get straight to the result that's expected, you don't necessarily need to draw out an entire Punnett square. That's just extra work. Okay, so if these are the expected genotypes for the F2, then what might I expect for my phenotypes? Well, if I look back at the parentals in the F1 generation, that might give me some ideas. So I might expect one red snapdragon to two pink snapdragons to one white snapdragon. Okay? And we can also do something similar if we think about crossing one of these F1 individuals with a white snapdragon or an F1 with a red snapdragon, right? So if we crossed big R with little r, then the first individual could produce two different types of gametes, right? Big R or little r. Second individual can only produce little r gametes. So then we'd expect one pink to one white if we crossed pink with white. And we'd expect something similar if we crossed the F1 pinks with red. We'd expect one pink to one red in the next generation. Okay, so here is the outcome from this cross. So we do indeed, in the F2 generation, wind up recovering exactly this. So one red or dark pink, two pink, intermediate pink, and then to one white. So yes, this trait does follow our expectations based on Mendelian inheritance. But it's different from what we've seen so far, right? So previously, we were only seeing two phenotypes. And in this case, we're seeing three phenotypes. So what we have here is a slightly different type of inheritance from what we have observed so far. So this is no longer simple dominant recessive inheritance. And instead, this type of outcome is referred to as incomplete dominance. Right? So there is an intermediate phenotype, pink, that is in between the phenotypes of the parents, right? So this can happen for a trait. Incomplete dominance is fairly common. So, again, I would encourage you to practice on drawing out other crosses to see what you'd expect from those crosses. Okay, so that's one alternative to simple dominant recessive inheritance. There's actually also another type of inheritance to be aware of that's called codominance. And so with incomplete dominance, we get an intermediate phenotype, whereas with codominance, if an individual has two different alleles, both of those alleles get expressed. And one of our classic examples of codominance is human blood types. So 3 out of the 4 blood types for people are, you can have type A blood, or you can have type B blood, or you can have type AB blood, right? So both A and B are expressed in someone who is type AB. But then you might ask, especially if this is you, well, what about type O? How does that fit into the picture? So although blood types are an example of codominance, they're actually a little bit more complex than that. So let's look at this in slightly more detail. So blood groups actually show a combination of dominance, or dominant recessive inheritance, and codominance. So the A and the B phenotypes are both dominant to O. Okay? But then the A and B phenotypes are codominant to each other. Okay. But then there's another kind of funny thing about blood types, which is this might lead you to think, okay, if A and B are dominant, then those are probably the most common blood types, right? AB or AB. But actually, most of the people in the US are type O. So they have the recessive phenotype. And that brings us to an important point that's going to come back to haunt us later on. And that is that Phenotype and frequency should not be confused with each other. So yes, type O is recessive, but it is also the most frequent blood type in the US. So just because an allele is recessive does not mean that allele is less common. It can be more common. So this language about inheritance, talking about incomplete dominance, codominance, dominant recessive, that does not tell you anything about the frequency of an allele in the population. And as I've said, we'll come back to that point. So now you might be really curious about how these blood types actually work. And it's kind of cool, and it builds on something we've already talked about. So it's a great time to discuss this further. So here's how they work. If you have the A allele, and for blood types, we typically write the A allele with a big I and then a superscript A. So if you have an A allele or two A alleles, then your body is going to produce a glycoprotein that winds up on the surface of your red blood cells. That's called the type A glycoprotein. So up here I've illustrated that with these little green triangles. So that is the type A glycoprotein. So if you have an IA allele, you will express this and it will show up on the surface of your red blood cells. Similarly, if you are type B, and that gets represented I with a superscript B, then you have a different type of glycoprotein that appears on your red blood cells membranes. And it's the type B glycoprotein that will show up, okay? So if you have one of these dominant alleles, that means that your body is able to produce either the type A or the type B glycoprotein. And in contrast, if you have type O blood, so type O is symbolized with a lowercase I, okay? And so if you are homozygous recessive for blood type, so your blood type is type O, then you don't make either of these glycoproteins. So your red blood cells would look something like this third picture. No glycoproteins present. And then lastly, if you are type AB, then you will have both the A and B glycoproteins present on your red blood cells. And I've noted on this slide that at this point it's now your job to list out the possible genotypes associated with each blood type. And as a hint, for a couple of these blood types, there is more than one genotype that will wind up producing that blood type. So I suggest writing out a list. Okay, so now here's the really cool part. Depending. So the glycoproteins that appear on the outside of a red blood cell, those are antigens if you don't already have them present in your blood. So if you don't normally have the type A glycoprotein in your blood, and you get blood that contains that type A glycoprotein, that's going to set off your immune system. And your immune system is going to attack those red blood cells as a foreign invader by creating antibodies against that protein. So, so just to kind of rewrite this, so we've got, if you have type A blood, you're going to make type A glycoprotein. And then if you give that to someone who has something like, let's say, give to someone who has type O blood, they don't have that glycoprotein normally present. So then that individual is going to produce anti-A antibodies, right? So against that A glycoprotein, anti-A antibodies, and the result will be agglutination. Okay, does that make sense? I hope it does. Think about it a little. So if you give type A blood to someone who normally has type B blood, same thing's going to happen. They're going to produce anti-A antibodies, and their blood will agglutinate, which is kind of a problem. Okay, so the way to think about this is if a red blood cell does not have that type of glycoprotein, then it's going to produce the anti-glycoprotein antibody in response to the presence of that glycoprotein, if you introduce that blood type. So what that means is, out of these blood types, there's one blood type that is a universal donor, and that universal donor is a blood type that doesn't have any glycoproteins on it, right? So type O blood has no glycoproteins. So, you know, if you could give someone with some type O blood to someone who's type A, someone who's type B, or someone who's type AB, and there is nothing to act as an antigen to trigger production of antibodies. And then at the same time, for example, if you have someone who has type AB blood, you can't give that blood to any of the other blood types, right? Because no matter what, that's going to trigger production of antibodies. You know, both anti-A, anti-B if you gave AB blood to someone with type O blood, anti-A if you gave that to someone with type B blood, and anti-B if you gave that to someone with type A blood. Okay, so think about that a little bit. It's kind of a complex idea, but it's fairly logical. So all things told, someone with type O blood is referred to as being a universal blood donor because anybody else can accept their blood. And then type AB blood is referred to as a universal receiver because someone with type AB blood has both of these glycoproteins present, so it's never going to produce, that person will never produce antibodies against their own red blood cells. Okay, so far, here are the three types of inheritance that we've considered. And in this case, we're thinking about traits that are determined by a single gene. So individuals will each have 2 alleles, right? So even with blood type, even though 3 alleles exist in people, each individual person will only have 2 alleles. Now the next type of inheritance we need to consider is called sex-linked inheritance. So this brings us back to thinking about this XY chromosome pair, right? And first we're going to talk a little bit about traits that are X-linked. And so these are traits that are found on this region of the X chromosome, which does not overlap with the Y chromosome. So if you are male, and you only have one X chromosome, you're going to be hemizygous for these traits. So they're located on this region of the X chromosome. So here's examples of some sex-linked traits. So one of them is red-green color blindness. So you may not have functional red or green cone cells in your retina. Another is hemophilia. So that's where individuals with hemophilia have a problem with a particular type of blood clotting factor. So they often require blood transfusions so that they don't bleed to death. That's also recessive. There's also a condition called Duchenne's muscular dystrophy, which is a degenerative muscular disease that happens in childhood. And then the last one here is called hypophosphatemia, where an individual winds up with a phosphate deficiency that can cause bow-leggedness. And just to give you some ideas about how you would write out alleles for these, right? So for red, green, color blindness, right? And if so, there are 2 possible alleles that can happen on the X chromosome. So X big C would be, you know, quote unquote normal vision. X little c would be colorblind. And this actually illustrates where it might be useful to use a letter other than C, because it's a little hard to tell a big C from a little c. And that would be fine. So you could do, X big B, X little b for color blindness. And then it's usually useful to also think about Y as being an allele, just as a placeholder, so that you remember that, to track what's going on with males. So for example, if we were thinking about a heterozygous female, she might be XB, big B, X little b. And then if we're thinking about a normal vision male, that individual would be X big B, Y. If we're thinking about a colorblind male, that individual would be X little b, Y. Okay? And the same concept for these other characteristics in general, right? So, you could use H's for hemophilia, D for muscular dystrophy. Hypophosphatemia would be a little bit different, right? So think about how you would write out alleles for hypophosphatemia. Okay, so now this brings us to a question. You might know that men tend to be colorblind much more frequently than women do. So why? And so if we just take a survey of colorblindness alleles in the US, somewhere around 8 out of 100 alleles, so 8 in every 100 X chromosomes, you will encounter one of these colorblind alleles. What that means is the probability of a man being colorblind, right, he's only got one of these alleles, is going to be 8 in 100, or 8%. And in contrast, for women to be colorblind, women would have to be X little b, X little b. So they would have to get 2 copies of this color blindness allele. So the chances of that happening are going to be, 8 in 100 times 8 in 100, which is about 0.6% of all females. So simply because males only have 1 copy of this allele, whatever copy they get is what shows up. They wind up being colorblind much more frequently than females do. Okay, and so we can kind of extrapolate on this and to think about sex linkage and probability in general. So overall, for any X-linked recessive condition, that condition is going to be more common in males than females. But in contrast, an X-linked dominant condition is going to be more common in females. So let's consider the case of hypophosphatemia here. So the probability of getting the hypophosphatemia allele is 1 in 10,000. So the chances that a male would wind up with hypophosphatemia are 1 in 10,000. But females have 2 opportunities, right, to wind up with that hypophosphatemia allele, right? If they're heterozygous, they're going to wind up with hypophosphatemia because they have 2 X chromosomes. And we're not going to do the full math for this, but overall what that means is their probability of getting hypophosphatemia is closer to 1 in 5,000. So, roughly double the probability for a male. And so now we come to kind of an interesting and I think kind of fun category of sex-linked trait, which are human Y-linked traits. So these are traits where the alleles are found on this part of the Y chromosome that doesn't overlap with the X chromosome, and they exist. So one example of a human Y-linked trait is a tendency to produce extra testosterone during development. And this later on leads to extra hair growth in an individual's ears. And I just think this is one of the coolest things ever. Here is a gentleman, he's a grocer in India who has one of these human Y-linked alleles. And so he has the world record for the longest ear hair. So if you have ear hair, you might as well grow it out and be proud of it. I think it's great. Okay, so, But coming back now to thinking about some genetic illnesses. So the vast majority of genetic illnesses are actually recessive. So similar to what I showed for color blindness. Why is that? Well, the reason why is if that illness were dominant, it would be expressed in both the homozygotes and the heterozygotes, right? So it would show up a lot. It would occur anytime that someone had that allele, it would be expressed, right? And what that means is that that allele would be subject to selection whenever it is expressed. And we're going to talk more specifically about selection later on. But what that means is if that genetic illness has implications for an individual's ability to survive and have kids, then anytime someone has that dominant allele, their chances of surviving and having kids are going to be reduced, and the chances of passing on that allele are going to be reduced. And in contrast, recessive alleles can wind up being hidden in heterozygotes, especially if they're fairly rare. And if an allele is not expressed, so if it's recessive, then it's not going to have consequences for whether that individual survives and has kids. So there's no opportunities for selection to act on that allele. And again, this is an idea that we're going to come back to later. But there's actually an interesting exception to that. And that exception is with Huntington's disease. So, and here's Willie Nelson. He actually died from Huntington's disease. And Huntington's disease is a genetic disease where individuals that have the dominant allele wind up having a mutation that adds sequences to their DNA. And eventually there's a buildup of protein in brain cells that poisons those brain cells and kills individuals with Huntington's. It's really tragic. So based on what I just told you, you might be wondering, how can Huntington's disease exist if it is dominant and always shows up in individuals and it kills them? And the answer is, this is a disease where the effects are really not seen until a person reaches middle age. So when they're younger, they don't really show characteristics of Huntington's disease. So it shows up after a point where someone has already had kids. And so that's part of how it's been able to be passed on. And this is a case where, we might think, well, maybe there's a role for some form of genetic counseling in a situation like this. It'd be really hard to discover that you're born with a dominant lethal allele like Huntington's disease. And if you knew about that at a younger age, that might wind up affecting some of the decisions that you make, with how you live your life. So a lot of implications to this. Okay. But I want to come back now to kind of wrapping up talking about the types of inheritance we've covered so far. So far we've just focused on traits determined by a single gene. So for all of these traits, each individual, if they're diploid, has two alleles. So we've looked at dominant recessive inheritance, incomplete dominance, codominance, and we've looked at both X-linked and Y-linked traits. So now in the next video, we're going to start stepping things up, and we're going to consider a dihybrid cross. So now we're going to start looking at two traits simultaneously. It's going to be fun.